In this module, we'll talk about meiosis. Meiosis is a type of cell division in which one cell gives rise to 4 daughter cells. Those 4 daughter cells have half the number of chromosomes or the genetic material as a parent cell. I would like to mention here that each of us has genetic contribution from both the parents. 50% of our DNA comes from one parent, the mother, and the other 50% comes from the other parent, the father. The chromosomes are in pairs, so we have 23 pairs of chromosomes, or in total 46 chromosomes. Here on the screen you can see the karyotype of a male person. Karyotype is basically arrangement of chromosomes in descending order by size of stained chromosomes. Here you can see that there are 2 chromosomes 1, 2 chromosomes 2, 2 chromosome 3s, and so on and so forth. So chromosome 1, we have 2 chromosome 1s. One came from father, the other came from the mother. If we have 2 chromosomes from 2 different pairs, from 2 different parents, it also implies that during the formation of the embryo or the fertilized egg, the cells that produce that fertilized egg or the embryo, the number of chromosomes in those special cells or the gamete cells, sperm or the egg, was also half. So how do we reduce the number of genetic material, the number of chromosomes or the genetic material in half? The trick is basically the DNA divide only once, whereas the cell divides 2 times. So in this process, we end up with products which have half the amount of DNA as compared to the parent. Let's look at the process. Meiosis 1 has a very long prophase. Prophase can be categorized into 3 different stages. Early prophase 1, it begins when the chromatin begins to condense. After the interphase, the chromatin that is condensing has already divided. The chromatin has divided during the S phase, which is part of the interphase. Also, in early prophase 1, the centrosomes have also divided and they are moving towards the opposite end of the cell. Here I would also like to mention that the nuclear envelope is still intact, like it was in mitosis, prophase of mitosis. The chromosomes in mid prophase, we will be able to, although in early prophase 1 we cannot see the chromosomes as discrete structures, in mid prophase, the next stage, we are able to see the chromosomes as discrete structures. Even at this stage, the nuclear membrane is still intact. The centrosomes are continuing their migration towards the opposite ends of the cell. Here you will see the important feature here is the homologs are pairing. Just as I told you that there are 2 chromosome 1s, the 2 chromosome 1s will pair up with each other. Each chromosome has already been divided. So each chromosome, individual chromosome, consists of 2 chromatids. Homologs, when they pair up, we have 4 chromatids lying next to each other, as you can see on the slide. In the next stage of prophase, the late prophase 1, we will see that the nuclear envelope now starts to disintegrate, and now the chromosomes will become available for connection with the kinetochore microtubules. The other important part of late prophase, as you can see on the screen, I will point it out also. Chromosomes, sister chromosomes, exchange parts. We'll look at this process in more detail. Here, for example, you can see part of the blue chromosome is now red, whereas part of the red chromosome is blue. I have erased the line so you can look at the same region. Chromosomes have exchanged pieces with each other. So because of this process, we can increase the genetic diversity. I'll explain that later, but right now we are talking about meiosis. We are going to talk about meiosis in 2 phases, meiosis 1, meiosis 2. I have just described the meiosis 1, 3 stages of prophase of meiosis 1. Let's continue and look at the other phases of meiosis 1. The next phase of meiosis 1 is, of course, metaphase. Metaphase 1 Here, like in mitosis we talked about, the chromosomes have lined up at the equatorial plate. Here now please notice that the chromosomes, when they line up together on the equatorial plate, the homologs are adjacent to each other. In mitosis, the homologs don't pair up. In meiosis, they pair up and they are lying next to each other. Here, the spindle fibers, they will connect to the kinetochore of the two homologs. Each homolog is going to go to the opposite end of the spindles. They are one chromosome 1 will go to one side of the cell, the other chromosome 1 will go to the other side of the cell. So the homologs are now separated in anaphase. Also, please notice that during the prophase of meiosis 1, these chromosomes also exchange different parts of each other. This process gives genetic diversity. Anaphase, as we have talked about in mitosis, similarly here, starts when chromosomes, in this case in mitosis, the sister chromatids were splitting apart from a single chromosome. Here in anaphase of meiosis 1, the homologs are splitting up and they're going to the opposite end of the poles. Telophase follows anaphase 1. In telophase, the nuclear envelope starts to reform, the chromosomes start to become decondensed, they start to loosen up and lose their shape. As I told you that there is a exchange of pieces, little fragments between the homologous chromosomes. Let's look at this process in a little more detail. Here you can see the two chromosomes, red and blue. Think of them as chromosome 1, like the one we saw in our karyotype, chromosome 1 cell from a human cell. So both the red and blue are chromosome 1s. They're homologous chromosomes. They are not identical, but they contain similar genetic information. So when these two chromosomes pair up, homologous chromosomes pair up, there's a process through which these two chromosomes can exchange pieces with each other. When this occurs, a point in the chromosome is created where two chromosomes are crossing each other. This is basically, you can see that under the electron microscope also. This is basically called the crossover point, and this results in two chromosomes which have now exchanged parts and they are no longer in their original condition or the parental condition. Next we will talk about meiosis 2. We'll continue with this process. We will talk about meiosis 2.